Chapter 8, Section 2 is on cell division, which is a process in which the cells are going to produce to make offspring cells. The main reason cells are going to divide is, one, the size is limited. We discussed in our cells chapter that the surface area to volume ratio is going to limit how large the cell can grow and still be able to take in enough nutrients to support itself. Another reason is to replace any damaged cells. And then the last one is just to grow. So we need to be able to produce more cells to grow. Prokaryotic cells divide a little bit differently than eukaryotic cells. Prokaryotes divide through binary fission, which is the process of splitting into two offspring cells. It takes place in about 3 stages. The first is their chromosome gets copied. Prokaryotes have a single circular ring of DNA, and they will need to copy that DNA in order to have a copy for each of the new cells. The cell is then going to grow to about twice its original size, and then once it has grown, it will split into 2. So like this cell over here was originally about this size, copied the chromosome, and then grew to about twice that size, and now you can see that it's pinching off in the middle where it will eventually split into 2 separate cells. Now, eukaryotes divide a little bit differently. They can divide through the mitosis process or growth division process, or through meiosis or reduction division. Now, with mitosis, the cell will divide to make 2 identical daughter cells. So if you start with a diploid cell, you will end with a diploid cell. With meiosis, you're going to start with a diploid cell and end with 4 haploid daughter cells. So we're going to be cutting the number of chromosomes in half. That's where this reduction division name comes into play. Now, the cell cycle takes place in a number of stages, but the stage that takes the longest is called interphase. Interphase begins about here and lasts the majority of the cell's life until about here. So you can see in this diagram, only a small part is where the cell is actually dividing. So there are 3 phases in interphase, G1, S, and G2, and then in cell division, that is made up of mitosis, or the M phase, where the nucleus will divide, and cytokinesis, where the cytoplasm will divide. Now in interphase, as I mentioned, this is where the cells spend the majority of their life in. This is just the time between cell divisions. And the 3 stages start with G1, which is where the cells are going to grow to their mature size. In the S phase, the DNA gets replicated. And then in the G2 phase, the cell is going to prepare for division, or mitosis. There's also a phase called the G0 phase, which is where cells are able to leave the cell cycle, usually when they're a mature size. Some of our cells that undergo this process are central nervous system cells, such as our brain and spinal cord. That's why if we have severe damage or trauma to those types of cells, a lot of times it can't be something that is fixed because there are not going to be new cells that are made. Those cells have already left the cell cycle. So the majority of our cells stay within this, following this life cycle pattern, but there are some that can go through that G0 phase where they leave the cycle. In DNA replication, the double helix gets split apart, so there's an enzyme that will come through and split all of these bases apart, and then each of these old strands will now serve as a template for building a new strand alongside it. So now each of the new cells that we produce will have a copy of our DNA. Here's an overall summary of mitosis. We just talked about interphase, and now we'll be moving into prophase, metaphase, anaphase, and telophase. That is the mitosis, or splitting of our nucleus stage. And then finally, we'll undergo cytokinesis, which is where these 2 cells will actually split apart. So in step 1 of mitosis, this is prophase, our chromosomes are going to start to form from the chromatin. So those histone proteins are going to be coiling up our DNA to form the chromosomes. You can see these rod-shaped structures beginning to form. The nuclear membrane starts to break apart. Centrioles move to the poles of the cell, and these spindle fibers will start to form. In metaphase, the spindle fibers will help to line up all of these chromosomes along the middle of the cell. This is called the metaphase plate. And this is usually where karyotypes are taken because all of the cells, all of the chromosomes are lined up neatly along the middle of the cell. In anaphase, the sister chromatids get pulled apart and moved to the opposite poles of the cell. And finally, in telophase, these spindle fibers will disappear. They're no longer used. They have already been split apart, or they have already moved the sister chromatids that have been split to the opposite sides of the cell. The nuclear membrane will start to reform, and the chromosomes will start to uncoil and go back to their chromatin form. So at this point, we have two nuclei that are in this cell that hasn't completely split apart yet. And finally, the last part is called cytokinesis, and this is where the cytoplasm will split. So in animal cells, the cell membrane will pinch in at the sides until it will eventually split. In plants, the Golgi bodies actually produce these little vesicles filled with cell wall material that will line up along the middle here until they're eventually all connected and form that cell wall. So the end result is you've got two new daughter cells. Here again is just a picture of the animal cell pinching off until it's split completely, and with the plant cell, the vesicles lining up along the middle until they're all connected to form that cell wall. Finally, the last part here is cancer. Now, cancer cells undergo uncontrolled growth. That's what cancer means, uncontrolled growth of cells. And it doesn't normally respond to the body's typical control mechanisms, which is why it can be so damaging. Mutations can actually interfere with the ability to slow down or stop this cell cycle. So instead of being wait and told to make new cells when they're needed, these cells can just keep replicating and replicating and replicating, which is why they can grow so quickly and spread so fast.